Good morning, student. I am Mr. Vinod Thombre, and we are studying the one of the topic from seed science, that is seed germination. The definition of seed germination: germination is the process by which the embryo wakes up from the state of dormancy and takes to active life. In simple words, germination can be defined as the growth of a seed into a young plant or a seedling. Germination is defined as the emergence of the radicle through the seed core. That means seed germination means emergence of plumule and radicals from the seed is called seed germination. The types of seed germination: there are three types of seed germination: epigeal type of seed germination, hypogeal type of seed germination, and viviparous type of seed germination. Let's see in detail. The first, this is the structure of bean seed, dicot seed. Here you will found seed coat, radicals in case of the structure of embryo, epicotyl, hypocotyl, and radical, and the food storage tissue called as cotyledon. First type of seed germination is epigeal seed germination. In this type of germination, the cotyledons are brought above the ground due to rapid elongation of hypocotyl. EP means above. That means when cotyledons they emerges out from the soil into the aerial part above the ground, and that type of germination is called epigeal germination. This type of germination is seen in many dicotyledonous plants such as bean, castor, sunflower, cucumber, etc. During this germination, the hypocotyl grows actively and become curved. It brings the seed above the soil. After coming above the surface of the soil, the hypocotyl straightens. The loosened seed coat fall down and the cotyledons become green. Initially, the green cotyledons they help for the photosynthesis. Now the epicotyl grows and plumule gives rise to green leaves, and the cotyledons fall down ultimately. This is the photograph which shows the difference between epigeal and hypogeal germination. In epigeal type of germination, we can able to see that the cotyledons that comes out above the ground surface. That's why it is called epigeal type of germination. And in hypogeal type of germination, the cotyledons remains present in a ground, that is below the soil. The next type of germination is hypogeal germination. In this type of germination, the cotyledons remains below the soil due to rapid elongation of epicotyl. It is found in many dicotyledonous seed and monocotyledonous seed. During this germination, the epicotyl elongates and become curved. It brings the plumules above the soil. Cotyledons remains present in the underground surface. In case of monocotyledonous seed like maize, the coleoptile, that is plumule covering, grows straight into the soil and comes out to form the green tube. Plumule elongates as well and comes out of the soil while contained in the coleoptile. The plumule ruptures the coleoptile with further growth. The coleorhiza covering of radicle along with radicle grows downward. After some time, coleorhiza ruptures due to the further growth of the radicle. The radicle forms the primary root, which is soon replaced by fibrous root. Here we see the type of hypogeal type of germination, where cotyledons they remain present underground the surface. Then the third type of seed germination is viviparous germination. In this case, during germination, seed that still attached to the parent plant and nourished by it. Vivipary generally occurs in mangrove plant. The mangrove plant that are generally medium-sized trees which grows in salty marshes of sea coast. The seeds of mangrove plant cannot germinate on the marshy habitat because of excessive salt concentration and lack of oxygen. The embryo of the seed continues growth while still attached to the parent plant. The radicle of the plant elongates considerably and projects out of the fruit. The lower part of the radicals becomes thick and swollen. Finally, dark dart light seedling breaks off the parent plant due to its increasing weight and gets embedded into the marshy in such a position that the plumule remains outside the saltish water. The radicle immediately forms new root and establishes the seedling as new plant. This is the special type of seed germination which is found in mangrove plant, because in coastline areas there is a excessive salt is present which does not allow the favorable condition for seed germination. Hence, in this case, the seeds that are germinated while they are attached to the parent plants. This is the diagram or figure which shows viviparous type of germination. Here we can able to see that the fruit that is still attached with the branch and elongates. Afterwards it fall downs and forms roots and develop into a entire plants. This is the type of germination. Now, what are the factors that affect seed germinations? That are several factors, external and internal factors. In external factors, water, aeration, temperature, salinity, and pathogenic organisms, they affect seed germination in different ways. In case of internal factor, seed dormancy is one of the important factor which affects the seed germination, because in case of dormant seed, the seeds which when placed in all favorable condition for germination, the seed does not show germination because of certain internal factor which causes the dormancy of seed. We can enhance this seed dormancy or break this seed dormancy with the use of gibberellic acid, and we can uh, able to germinate the seed with the treatment of gibberellic acid. The another internal factor is viability of seed. That means the ability of embryo to germinate and grow into a seedling, and after maturation that forms the entire plant. The viability of embryo that affects the growth of and germination of seed. So those are some factors, external and internal factors that affect the seed germination. Thank you.